Okay, class, so we're going to start with part two here, and part two really builds on part one, and we're going to look at sort of more specifics on sort of bidirectional transport and how secretory proteins go through the secretory pathway, and then we're going to look at a little bit of what happens once a vesicle reaches the Golgi and what happens to the cargo proteins when it gets to the Golgi. So that mainly involves glycosylation. Okay, so like I said, in this part of the lecture, we're going to really focus on the bidirectional transport between the endoplasmic reticulum and the Golgi apparatus. And two terms you need to know is sort of forward transport, which is called anterior grade transport, and retrograde transport, which is the retrieval of things from the Golgi to the endoplasmic reticulum, right? And this is sort of serves as a recycling mechanism, as you'll see, right? Cargo goes forward, and anytime an ER resident protein or something that is needed for this process of fusion will then actually get recycled back. Okay, so it's a recycling step. Okay, so let's look at exit from the ER. Okay, this is all mediated by the COP2 vesicles, and we saw how those formed last time. You have your SAR GTP in Sec 2324 and Sec 1331, and these are sort of selecting cargo receptors as well as cargo. Okay, now in this whole thing, in the ER, there are actually ER resident proteins that help in folding, and we've already seen a couple of these when we were looking at translocation. We saw the protein BIP. We'll see it again in a second. And we also saw when you start to form disulfides, you need protein disulfide isomerase, and there's also other proteins that are needed for folding and early glycosylation that are not secreted proteins. So those are ER resident proteins. And so you have cargo that's actually actively selected, and then ER resident proteins that are excluded, okay, and not included in the vesicles. And here's an example of sort of how that happens and the ER quality control. So this is an antibody. And so antibodies are actually 4 proteins. And so you have 2 heavy chains, which is shown in dark green here, and 2 light chains, which are shown in light green. And so here you have BIP. This is a HSP70 family member, and it's binding to sort of regions on the antibody heavy chain. And the 2 heavy chains will come together, and then you'll get formation of the light chains. And once the full antibody is made, it's sort of released from BIP, and then it actually gets incorporated into a vesicle so that it can be secreted. Okay, and all secreted proteins for the most part sort of follow this path where you actually fold in the ER, and then once you're properly folded, you're sort of released and then sort of selectively incorporated into a budding transport vesicle, while ER residents are actively retained. Okay. Once the vesicles bud off the endoplasmic reticulum, they fuse, and they undergo this thing called homotypic fusion. Okay, where you have 2 uncoated COP2 vesicles, and then you have an old trans snare complex that's there, and then the protein NSF comes and dissociates the trans snare pair, so then you can reform the trans snare pair, and then you can get homotypic fusion where these former COP2 vesicles start to fuse together. Okay, and all the snares are synthesized on the ER, and so when this happens, they actually form the trans snare, and then they fuse, and eventually when these get to the Golgi, they'll get retrieved and recycled back to the endoplasmic reticulum. And so when you have this homotypic fusion, you form this thing called a vesicular tubular cluster. Okay, and here's just a nice EM of the vesicular tubular clusters. And here you can see vesicles budding off of the endoplasmic reticulum, and they'll start to then fuse together to form these vesicular tubular clusters. Okay. For graduate students, you may come across it as called the ergic, for ER to Golgi intermediate compartment. In the literature, it's also called that. But in general, vesicular tubular clusters are sort of the more common name for them. Okay. So once again, there's actually retrieval, right? The trans-snare pair that has mediated the fusion event actually gets recycled. And so what you have is your endoplasmic reticulum, and the COP2 proteins are forming the vesicles. Once the coat is shed, the COP2 vesicles fuse to form the vesicular tubular cluster, and then things like the trans-snares are actually recycled and retrieved back to the ER, and this is mediated by COP1 vesicles. Okay, so COP2s are involved in anterior grade transport. COP1s are involved in retrograde transport. Other proteins that are retrieved are also the cargo receptors that are needed to get proteins from the ER to the Golgi, as well as any ER resident protein that is inadvertently gotten packaged into a vesicle. Those proteins, like the ER chaperones and things, will also get retrieved, and we'll see how those ER resident proteins get retrieved in the next slide here. Okay, so here's the retrieval. So let's say you have an ER resident protein like BIP, or like protein disulfide isomerase, PDI. They all have a signal sequence on them called KDEL. So if you think back to last lecture, I told you guys that there was a table and you didn't have to memorize the signal sequences for anything except for one, and that one was the KDEL sequence, okay? And it's just a simple thing to remember. Lysine, aspartic acid, glutamic acid, and leucine, okay, KDEL. This sequence is present on every ER resident protein, every soluble ER resident protein. So in the event that an ER resident protein gets inadvertently packaged into a COP2 vesicle, and that vesicle then goes on to fuse to form the vesicular tubular cluster, and which eventually will go on to become part of the Golgi. If that protein has a KDEL sequence, it will be bound by the KDEL receptor, and then that actually gets bound and packaged into a COP1 vesicle and retrieved back to the endoplasmic reticulum. And that way the cell doesn't have to continually make its soluble ER resident proteins. So you have sort of a twofold thing happening here. You have cargo actively being selected into a COP2 vesicle going forward, and any ER resident protein that has somehow gotten inadvertently packaged into a COP2 vesicle, when it fuses and becomes the vesicular tubular cluster, or the cis-Golgi, or becomes part of the cis-Golgi network, a COP1 vesicle will take the KDEL receptor bound to the KDEL containing ER resident protein, and it'll form a retrograde vesicle that then goes back to fuse with the endoplasmic reticulum, and thereby retrieving the thing. So once again, there's a molecular mechanism that I would like you guys to know. This one's actually quite simple. You just have your KDEL receptor, and that's packaged into a COP1 vesicle to be brought back to the endoplasmic reticulum in the event that a soluble ER resident protein leaks out. Okay. So here's more of a global overview of that. You have here these sort of square yellow boxes as secretory proteins and the ER residents, and so as well as sort of the KDEL receptor, and the KDEL receptor wants to reside on the Golgi in the cis-Golgi network, as well as the vesicular tubular cluster. So these COP2 vesicles form, they undergo homotypic fusion in forming of the trans-snares, and then in the event that an ER resident protein leaked out, the pH difference causes the KDEL receptor to bind to KDEL containing proteins, and then those are packaged into COP1 coated vesicles and are brought back in a retrieval process. Meanwhile, if you sort of follow these diamonds and squares going forward, they're just secreted proteins that are going to sort of transverse the Golgi, and as they're doing this, all of the glycosylation gets modified, which we'll see in a second. Okay, so here's just a 3D model of the Golgi apparatus. You have your trans-Golgi network, and then your trans, medial, and cis, as well as your cis-Golgi network here. And one of the things with the Golgi, which is kind of cool, it's always capitalized. It's one of the few subcellular compartments that's capitalized, because the guy who discovered it was Camillo Golgi, and that was his last name. He was an Italian. But this is just the Golgi, and the big thing that happens here in the Golgi is that all of the glycosylation that was started in the endoplasmic reticulum gets further processed. Okay, so all of the oligosaccharides on proteins get modified in the Golgi. Okay, and what's occurring in the different stacks here is quite specific. Here's just some images of the Golgi and the vesicular tubular cluster, as well as you can see the rough ER here with the ribosomes bound, and you have your nuclear envelope. So it sort of resides very close to the nucleus. Okay. Here's just another image of it, your cis-Golgi network, and then your cis, trans, medial, and then your trans-Golgi network sort of out at the end. If you stain it, you can see here's the nucleus, here's sort of the outer plasma membrane of the cell, and here's the Golgi sort of right there, and we saw it before, and the Golgi is just sort of stained. One of the things that I didn't mention back here, but you actually have motor proteins that move them around, and we saw this on the video on the first day of class, where you saw the motor proteins sort of moving things forward in this whole process. Okay, here's the Golgi stained in a plant cell, pretty similar, right around the nucleus for the most part. Here's just some more images of the Golgi, and once again, the Golgi stacks are distinct depending on the oligosaccharide modifying enzymes. You can tell if you're looking at the cis medial or trans Golgi, or the cis Golgi network or the trans Golgi network. So here's the cis face here, and this is the trans Golgi network. Okay, and here's the cis Golgi network over here, and then sort of the medial Golgi is in the middle there. So here's what's happening in the Golgi. So you get vesicles and vesicular tubular clusters from the ER, and then you get phosphorylation of the oligosaccharides for lysosomal proteins, and then in the cis you get sort of modification, removal of the mannoses, addition of glucnac, and addition of Gal and Nana, and then other sulfonation reactions of tyrosines and carbohydrates can happen in the trans-Golgi network. I'm a little less concerned that you guys memorize all of these, but it is really important to understand that there is processing of the glycosylation through the Golgi, okay? And depending on how a specific protein is processed, it will then determine whether or not it's headed to the lysosome, the plasma membrane, or into a secretory vesicle. Here's sort of a famous secreting cell. It's the goblet cell, sort of shaped because it looks a little bit like a goblet. And these are in the intestine, and they're involved in sort of secreting mucus, proteoglycans and stuff like that, glycoproteins, and you have these huge secreted vesicles that secrete mucus from the apical surface of the cell. There's your Golgi down here. Okay, and just sort of note these large secretory vesicles that have sort of come off the Golgi. Okay, and they're just waiting to be fused with the plasma membrane and release their constituents. Okay, so glycosylation and the modifications of glycosylation that's happening, you actually have a couple different types of glycosylation. There's two types of end-linked glycosylation. You can have a complex oligosaccharide, and here are the different sugars N-acetyl acid is red. Galactose is sort of orange here. Mannose blue, and then N-acetylglucosamine or glucnac is in yellow here. Okay, so this is sort of the core region, and then these get modified as proteins go through. Once again, no need to memorize these details, just know that these different sugar groups are being added as secretory proteins sort of travel through the Golgi apparatus. And here's sort of what happens. So in the ER lumen, you first get the end-link glycosylation, and then you have the glucose trimming. Glucose is in green here, and that's done by glucosidase. We looked at that last time during the whole protein folding. And then a couple of the mannoses get cleaved off by the ER mannosidease. Okay, then the protein gets packaged into a vesicle and starts to go to the Golgi, and then other sort of Golgi mannosidases will cleave cleave off some of the mannoses, and then you have the N-acetylglucosamine transferase, which adds glucnac, and then you also can get other sugars added like nana and glucose and other things, and galactose, I mean, not glucose, here as it sort of travels and gets sort of towards the trans-Golgi network. Okay. You can also have O-linked glycosylation. So normally N-linked is linked with the nitrogen. So here you have N-acetylglucosamine linked to asparagine, but you can also have glycosylation linked to the threonine, and that's considered O-linked. Okay, N-linked through the N, O-linked through an O. Okay, and O-linked tends to occur more on proteoglycans. And last lecture I sort of told you the difference between proteoglycans and glycoproteins, if you remember. If you don't, you can go back and rewatch the last lecture. You can also have sulfonated proteoglycans, so that's just the addition of sulfur groups onto it. And once again, these are sort of found in mucus and also in the extracellular matrix, which we'll cover in a few lectures. Here's the structure of N-linked glycosylation, your glucnacs. There's your asparagine, your mannoses, and here's sort of a space-filling model of it. Okay, and this all helps with protein folding and also makes secreted proteins soluble to, or resistant to proteolysis. And we saw in last lecture the staining of sort of the carbohydrate layer of proteins out on the plasma membrane. So almost all secreted proteins get glycosylated in some form or another. Okay, so one last thing I want to share with you. I think this is the last slide. This is the concept of vesicle transport versus cisternal maturation. And I don't know what the current status of this in the field, but it's sort of interesting to think about, okay? So in vesicle transport model, what it says is the Golgi stacks are just incredibly static, okay? The cis is always the cis, the medial is always the medial, and the trans is always the trans. So you have your cis, medial, and trans Golgi, and those are just static. And what you have are vesicles that butt off and fuse, and then in between these individual stacks, you have bidirectional transport where each vesicle is butting off and fusing with the next. So things that are in the cis-Golgi network will butt off and fuse with the cis-Golgi, and then vesicles will butt off the cis-Golgi and fuse with the medial Golgi, and then things from the medial Golgi will fuse with the trans-Golgi. You get the idea. And meanwhile, things that are needed in each of these compartments are recycled back. And so you have one vesicle set going forward and one vesicle set going backwards. And that's the vesicle transport model. Very static membrane stacks within the Golgi. Now, cisternal maturation model is a bit different, and it's more like an assembly line, okay? And so what it says is you get vesicles budding off of the ER, these fuse to form the vesicular tubular clusters, and then retrieval alone will make this vesicular tubular cluster look more like the cis-Golgi network, okay? And then as you sort of go through, that cis-Golgi network will then maturate to become the cis-Golgi, and there's no forward vesicle budding. It's only retrograde transport, where things that make the cis-Golgi is because everything that became in the medial is retrieved back to the cis-Golgi, right? And same thing with the trans. As the medial maturates to become the trans, it does so because everything that's needed in the medial Golgi is being retrieved from the trans Golgi, okay? Until you finally get to the end at the trans Golgi network, where vesicles bud off and go forward. So I really like this cisternal maturation model. But this one is also, there's a lot of evidence for it. It's not really clear which is really happening. And, you know, sort of the way I like to think about science is if there's sometimes two really good competing models with a bunch of evidence supporting both, oftentimes the thing to do is to sort of believe both models. Okay, so this is something I really hope you guys can appreciate and sort of understand. You know, this is one of those complex ideas that you know, I think I would like you guys to be able to explain the differences between these and whatnot. Okay, so that's it. So I didn't update this last slide. I apologize. This should say for Tuesday, your next exam.